everyone. Welcome all to a new session on molecular biology. We all know that genetic information of every living being is stored in its nucleic acid. Now we are going to see the way in which the genetic information is stored, read and executed. Genetic code and mechanism of translation. We already know the purpose of DNA chromosomes as carrying the information for building the proteins of the cell and the RNA as the intermediary for doing so. How is it possible a molecule made up of just 4 different nucleotides joined together can tell a cell which of the 20 odd amino acids be joined together to form a functional protein? First of all, we must understand the genetic code and its features. This genetic code is a pattern of storing genetic information. It is an arrangement of nucleic acids on mRNA which is translated to amino acid sequences. Translation, as the word implies, converting something like a code or any unknown language to an understandable format. The amino acid sequences makes sense. They are in fact modified to fully functional proteins. Now, let us deeply study about genetic code. Francis Harry Compton Crick, a British molecular biologist, played a crucial role in research related to revealing the genetic code along with Sidney Brenner. He is widely known for the use of the term central dogma. The central dogma of molecular biology states the flow of information is unidirectional. Discovery of genetic code. Genetic code is the nucleotide base sequence on DNA and subsequently on mRNA by transcription, which will be translated into a sequence of amino acids of the protein to be synthesized. The code is composed of codons. Codon is composed of 3 bases, for example, ACG or UAG. Each codon is translated into one amino acid. Deciphering the genetic code requires determining how 4 nucleotides such as adenine, thymine, guanine and cytosine could encode more than 20 amino acids. Since there are 20 different amino acids that occur in proteins, there must be more than 20 codons to include signals for starting and stopping the synthesis of particular protein molecules. The 4 nucleotide bases such as A, G, C and U in mRNA are used to produce the 3 base codons. A single base cannot be a codon because for 20 amino acids we have only 4 bases. Pairs of bases cannot also be a codon because there are only 4:2 or 16 possible pairs. Francis Crick and Sidney Brenner determined that the DNA is read in sets of 3 nucleotides for each amino acid. Triplets of bases are possible because there are 4:3 or 64 triplets, which is more than enough. There are therefore 64 codons which code for the 20 amino acids, and since each codon codes for only one amino acid, this means that there are more than one code for the same amino acid. Marshall Warren Nirenberg, an American biochemist and geneticist, identified the codons that specify each amino acid. Nirenberg's initial goal was to determine whether DNA or RNA copied from DNA was a template for protein synthesis. Experiments with synthetic RNA. Nirenberg and Heinrich Mattei, a postdoctoral fellow from Germany, began their experiments by studying DNA and RNA molecules. In DNA, the nucleotides are adenine, guanine, cytosine and thymine. In RNA, uracil replaces thymine, the basic molecular unit that are strung together to form long linear molecules of DNA and RNA. Nirenberg and Mattei set out to create a cell-free environment that would let them examine how the minute mechanisms of RNA actually work free of the normal biological processes of cells that could obscure molecular activities. Cell-free environment. A cell-free environment is created when cell walls are broken down, releasing the cell's contents. The remaining cytoplasm can still synthesize protein when RNA is added, allowing the researchers to design experiments to determine how RNA works free of the complicated biological processes that could shroud molecular activity. After working with several possible organisms to serve as models, they selected Escherichia coli, a common bacterium found in the human colon. They added the E. coli extract to 20 test tubes, each containing a mixture of all 20 amino acids. In each test tube, one amino acid was radioactively tagged, a different one in each test tube. The reaction was observed by monitoring radioactivity. Incorporation of a hot amino acid would form a hot protein. The poly-U experiment. Synthetic RNA made up of only uracil units were added to each of the 20 test tubes containing mixture of all 20 amino acids, and the one containing phenylalanine showed unusual activity. Nirenberg and Mattei concluded that traces of uracil had directed the synthesis of phenylalanine. Synthetic RNA made up of multiple batches of 3 units of uracil directed the synthesis of an amino acid chain composed of only phenylalanine. One 3-unit batch of uracil could be read as UUU or poly-U, which was a 3-letter shorthand method or code word for identifying phenylalanine. They recognized the poly-U as the messenger. Their experiments proved that messenger RNA, which transcribes genetic information from DNA, directs protein synthesis. Nirenberg and Mattei concluded that synthetic RNA made up of a chain of multiple units of uracil instructed a chain of amino acids to add phenylalanine. The uracil chain or poly-U served as a messenger directing protein synthesis. The experiment proved that messenger RNA transcribes genetic information from DNA, directing the assembly of amino acids into complex proteins. Thus, the key to breaking the genetic code, molecular biology's Rosetta Stone, had been discovered. After Nirenberg and Mattei discovered the first word of the genetic code, scientists were trying hard to translate the unique code words for each amino acid, hoping to read the entire genetic code of living organisms someday. Following the poly-U experiment as a model, Nirenberg and his colleagues recognized the nucleotide combinations for the incorporation of other amino acids. The codes for other amino acids were identified, for example, AAA for lysine and CCC for proline. One unit of a triplet code was replaced with another nucleotide yielding a different amino acid. In one example, synthetic RNA containing 1 unit of guanine and 2 of uracil caused incorporation of valine to a growing polypeptide chain. In 1964, Nirenberg and Philip Leder, a postdoctoral fellow at NIH, discovered a way to determine the sequence of the letters in each triplet word for amino acids. By 1966, Nirenberg had deciphered the 64 RNA 3-letter code words for all 20 amino acids. The language of DNA was now cracked and the code was expressed in a chart. Introduction of the word codon. In the late 1950s, the biochemist Sidney Brenner coined the term codon to describe the fundamental units engaged in protein synthesis, even though the units had yet to be fully determined. Francis Crick popularized the term in 1959. After 1962, Nirenberg began to use codon to characterize the 3-letter RNA code words. With one of each of the 4 nucleotides occupying a place in a 3-letter codon arrangement, Nirenberg quickly deduced that there were 64 possible combinations of 3-letter codons. In 1964 and 1965, Nuremberg's postdoctoral researcher Philipp Leder developed a sophisticated filtration machine that helped the team determine the order of the nucleotides in the codons. This development speeded up the process of assigning code words to amino acids. By 1966, Nuremberg deciphered all the 64 RNA codons for all 20 amino acids. Rules governing genetic code. The genetic code is subject to 3 rules that govern the arrangement and use of codons in the messenger RNA. 1. Codons are read in a 5' to 3' direction. 2. Codons are non-overlapping and the message contains no gaps. 3. The message is translated in a fixed reading frame which is set by the initiation codon. Reading frame is a series of nucleotides read in sets of 3 codons. Only one reading frame is correct for encoding the correct sequence of amino acids. Most amino acids have more than one codon. Only methionine and tryptophan have a single codon. Also, multiple codons corresponding to a single amino acid usually differ only by the 3rd base. For example, GGU, GGC and GGG all code for glycine. This situation is called degeneracy, that is, the code is degenerate. Start codon. AUG codes for both methionine and the start of translation depending on the circumstances. In eukaryotes, the start codon codes for methionine, while in prokaryotes it codes for formilated methionine. There are also alternative start codons in both eukaryotes and prokaryotes. They still code for methionine and only the initiator tRNA is different. For example, CUG in eukaryotes, GUG, UUG, AUU and CUG in prokaryotes. Termination, stop or nonsense codons. 3 of the 64 codons UAA, UAG, UGA do not code for any amino acid. They are termination codons which, when one of them appear in an mRNA sequence, it indicates finishing of protein synthesis. They are also called stop codons dedicated to ending translation. The 3 stop codons also have colloquial names. UAA stands for ochre, UAG for amber, UGA for opal, with UAA being the most common in prokaryotic genes. Characters of the genetic code. First one, specificity. The genetic code is specific, that is, a specific codon always codes for the same amino acid. Second one is universality. The genetic code is universal, that is, the same codon is used in all living organisms, prokaryotic and eukaryotic. The third one is degeneracy. The genetic code is degenerate, that is, although each codon corresponds to a single amino acid, one amino acid may have more than one codon. For example, arginine has 6 different codons. Wobble hypothesis. There is a phenomenon called wobble in which a codon-anticodon match is allowed and stabilized for translation even if the nucleotide in the 3rd position is not complementary. Many amino acids are specified by more than one codon. It is also known as redundancy. Frequently, a tRNA can translate more than one of these codons, sparing the cell from making multiple tRNAs to carry the same amino acid. For instance, the tRNA for arginine can translate both the CGA and CGG codons that specify arginine. Correct base pairing is required at the 1st position of the codon, that is 3rd of anticodon, and the 2nd position of the codon or 2nd of anticodon. The third position of the codon does not always need to be paired with the anticodon. For example, it is allowed to wobble in some cases. In most instances, the third base is the degenerate base. It is sometimes referred to as the wobble base. In 1966, Francis Crick devised the wobble concept. It states that the base at the 5' end of the anticodon is not as spatially confined as the other two, allowing it to form hydrogen bonds with more than one bases located at the 3' end of a codon. It is not necessary that a specific tRNA anticodon would exist for every codon. Highly purified tRNA species of known sequence could recognize several different codons. An anticodon base was not one of the 4 regular ones, but a 5th base, inosine. The wobble rules. 5' anticodon base G can pair with U or C at the 3' codon site. 5' anticodon base C can pair with G at the 3' codon site. 5' anticodon base A can pair with U at the 3' codon site. 5' anticodon base U can pair with A or G at the 3' codon site. 5' anticodon base I can pair with U, C or A at the 3' codon site. Gene mutation. Altering the nucleotide sequence leads to changes in the codons and thereby may even alter the amino acid sequence too. Changing a single nucleotide base on the mRNA can lead to any one of the following 3 results. The first one is silent mutation. The codon containing the changed base may code for the same amino acid. Second one is missense mutation. The codon containing the changed base may code for a different amino acid. The third one is nonsense mutation. The codon containing the changed base may become a termination codon. Decoding the genetic code. The decoding of the codons on the mRNA to a meaningful amino acid sequence is possible by the help of a tRNA molecule and a set of enzymes called amino-acyl tRNA synthetases. 3 regions of each tRNA molecule are used in the decoding of mRNA. They are anticodon, amino acid attachment site and recognition region. The 3-dimensional structure of tRNA molecule was first determined through X-ray crystallographic studies by Alexander Rich and Aaron Klug in 1974. For each codon in the mRNA that specifies an amino acid, there is one tRNA molecule containing a complementary sequence of 3 adjacent bases that can pair with the codon on mRNA. The correct amino acid is attached to the other end of the tRNA. When it identifies the appropriate anticodons, it comes and attaches to the mRNA, which initiates the translation process. Translation. Translation can otherwise be called as protein synthesis. It is the decoding of the genetic code into the amino acid sequences. It is the second step in central dogma. It describes how the genetic code is converted into amino acid. There are 3 important steps in translation. First one is initiation. During initiation, the mRNA, tRNA and amino acid all come together within the ribosome. The initiation site is the start codon. The start codon marks the beginning of coding sequence of a gene. It specifies methionine that is brought by tRNA that bears the anticodon UAC for AUG according to the complementary base pairing rule. Requirements of initiation. The first one is charged tRNA. Every tRNA has an amino acid binding site opposite to its anticodon arm. The tRNA needs to be charged to start the process of translation. Charging of tRNA occurs when the amino acid specific to its anticodon attaches to its amino acid binding site. The amino acid is carried at the 3' hydroxyl end of the tRNA molecule. The charged tRNA is called amino acyl tRNA. tRNA is charged by forming ester bond between the COOH group of the amino acid to the 2' or 3' OH group of the adenyl group at the end of the acceptor arm, as shown in the following figure. The charging of tRNA is catalyzed by the enzyme amino acyl tRNA synthetase. The next one is ribosomal subunits. Ribosome is a site where translation takes place. Both the small and large subunits assemble along with the mRNA and other initiation factors to start the process. This figure shows the 3 different sites in these ribosomal units. The next requirement is mRNA. In mRNA, there are start codon and stop codon. There might be many AUG in a single mRNA. The right AUG is picked up by the ribosome. The recognition mechanism is different in prokaryotes and eukaryotes. In prokaryotes, the recognition site is Shine-Dalgarno sequence, whereas in eukaryotes, it is the 5' GCAP. The codons on mRNA are read in 5' to 3' direction, and the anticodons on tRNA binds to the codons on mRNA in an antiparallel fashion. That is, its 5' base hydrogen binds to the mRNA's 3' base. The first 2 bases from the 5' to 3' direction are constrained bases in the codon. It is vital for amino acid insertion, whereas the third one at the 3' end is not necessarily be a perfect match for an amino acid to be added successfully to the chain. It is explained before in Wobble concept. The next process is elongation. During the elongation step, amino acids are added to the carboxyl end of the previous amino acid. The polypeptide chain grows as the ribosome moves from the 5' end to the 3' end of the mRNA. tRNA with anticodon complementary to second mRNA codon binds to ribosomal A site with appropriate amino acid attached to it. A peptide bond is formed between the first and second amino acid in P site and A site respectively. The formation of peptide bond is catalyzed by the enzyme peptidyl transferase present in the ribosome. The energy required for this process is provided by the hydrolysis of GTP. Now, the first tRNA in P site leaves the ribosome as it is detached from the amino acid. As the ribosome moves along the mRNA, the tRNA with second amino acid moves to P site, exposing the A site. The A site is now free for the amino acyl tRNA corresponding the next codon to bind to it. Then, as before, a peptide bond is formed between the second and third amino acid, and the ribosome moves along a single codon, leaving the A site free for the next anticodon. This process is repeated until the ribosomes encounter a stop codon. The next step is termination. When the A site reaches a stop codon, instead of tRNA, release factor proteins binds to the A site. There is no tRNA available with the anticodon for stop codons such as UAA, UAG and UGA. The binding of release factors causes the release of newly synthesized polypeptide and mRNA from the ribosome complex. Finally, the ribosomal subunits also get separated from each other. Efficiency of translation. 2 mechanisms significantly increase the overall rate of translation. The first one, a single mRNA molecule is translated by many ribosomes simultaneously. Electron micrograph and sedimentation analysis have proved this phenomenon. It showed mRNA attached to multiple ribosomes bearing a nascent growing polypeptide chain. This structure is named as polyribosomes or polysomes. The next step is rapid recycling of ribosomal subunits after they dissociate from the 3' end of mRNA. The poly A tail of mRNA is found close to the 5' end. Poly A binding protein can interact with both mRNA poly A tail and the 4G subunit of yeast EIF4. The 4E subunit binds to the 5' end of mRNA. As a result, the two ends are joined to form a circular mRNA. Thus, the ribosomal units that disengage from the 3' end are positioned near the 5' end, facilitating the reinitiation. Inhibition of translation. Translation is inhibited by certain factors called translation inhibitors. They stop or slow the proliferation and growth of cell by disrupting the processes that lead directly to the generation of new protein. Antibiotics are a good example for translation inhibitors like streptomycin, tetracycline, chloramphenicol, etc. Let us now summarize what we have learned in this session. Genetic code is the nucleotide sequence on DNA which will be transcribed to mRNA and afterwards translated to protein. It carries information necessary for the expression of a gene to protein. A single codon consists of 3 nucleotide base which represents a single amino acid. Any alteration in the nucleotide sequence leads to the changes in the codon and thereby can sometimes alter the total protein. Translation is the process by which the genetic code transcribed on mRNA is converted to amino acid sequences. It requires the participation of ribosomes, tRNA, catalytic enzymes and many other factors. The whole process is divided into initiation, elongation and termination. The mechanism differs in prokaryotes and eukaryotes by the difference in the ribosomal units and factors involved. Here are a few questions for you to work out. 1. Explain poly-U experiment in deciphering the genetic code. 2. How does an amino-acyl tRNA synthetase recognize the initiation codon during gene expression? 3. What are the different sites on ribosome? Explain its function. 4. How many energy-rich molecules are utilized during the addition of one amino acid to a growing polypeptide? 5. Explain Waubel hypothesis. Here I can suggest a few books for your reference. Molecular Cell Biology, 5th edition, written by Lawdish H., Burke A., Matsudaira P., Kaiser C., Krieger M., Scott M. P., Sipersky L. and Darnell J. Published by Freeman W. H. and Company, 2003. Genes 8, written by Levin B., published by Pearson Prentice Hall Publication, USA. Biochemistry, 5th edition, written by Burge J. M., Timosko J. L., Stryer L., published by Freeman W. H. and Company, New York. Cell and Molecular Biology Concepts and Experiments, 7th edition, written by Karp G., 2013, Wiley Publication, USA. Here I can suggest a few websites for your reference: www.powershow.com, www.austincc.edu. Thank you for watching this program. Let's meet in the next session with another topic. Until then, bye.